Well, hello, everyone, and welcome to the Cell and Gene Therapy Virtual Event Series 2024, brought to you by LabRoots. We are thrilled that you are here, and we have lots in store for you today. Today's first keynote presentation is titled Analytical Challenges and Solutions for Enabling Nanoformulations and Non-Viral Modalities for Next-Generation Therapeutics. I am Israel Gonzalez of LabRoots, and I will be your moderator for today's webinar. Before we begin, I would like to remind everyone that this event is interactive, and we encourage you to participate by both communicating with each other in the peer-to-peer chat and also by submitting questions during the presentation for our speaker to answer after. Now let's go ahead and get started. I would like to introduce our speaker today, Jelle Penders, PhD, CEO, CTO, founder of SPARTA Biodiscovery. You can view our speaker's complete biography on your screen. Now, Dr. Penders, you may now begin your presentation.
Great, thank you very much, Israel, and great to have everyone here today. I saw there's people from all over the world, from very early in the morning to late in the afternoon. So it's my pleasure to talk to you today about basically my favorite topic, analytical challenges for nanoformulations, and of course the solutions that are being developed across the world for this. So today I'm going to give you of these analytical challenges in the next 25 minutes approximately, and this really aimed to give a broad overview of what has been done and what's out there, and for those that may not be familiar with that, a good introduction, and for those that are familiar with it, hopefully some insights from our perspective. So I'm just going to tell you very briefly who I am, so you also know why I'm speaking and what I'm coming from. So I'm originally from the Netherlands. I've studied in a whole range of European countries and a short stint in the US as well. And my background is really in chemistry, analytical chemistry, everything to do with nanomaterials and nanoanalytics. I always found very, very interesting. I've used a lot of the techniques that I'm going to present as well, mainly spectroscopy-based techniques and electron microscopy. And this led to development of a new technology, which is the basis of our company, SPARTA, which I'll touch on a little bit at the end as well, and from which I'm currently the CEO and co-founder as well. So there's just a little conflict of interest disclosure here as well, that I set up a company and lead a technology in this space. Great. So first off, what are nanoformulations? What is nanomedicine, and why is this interesting, and why is this important? So nanomedicine is really the delivery of small molecule drugs, or more recently, more importantly, nucleic acids through a small carrier in the nanoscale, rather than delivering set molecules, for instance, as active ingredients directly into the body. And this is done because it gives a lot of different advantages over traditional systems, mainly to improve the safety of delivery of some of these molecules, improve the accuracy and efficacy as well of these agents. And this has really been an enormous revolution in pharma that has started probably around the 50s or 60s of the 1950s or 1960s, of course. But really, we see enormous breakthroughs in the last decade or so. Particularly, culmination of that, of course, was the COVID-19 vaccines delivered in very record time. So what we've seen now is this field growing from a very academic pursuit to something that basically every pharma company in the world has a program towards, be it viral delivery or non-viral delivery, or both. And we see a lot of very exciting young companies in this space, as well as, of course, still very much ongoing worldwide academic efforts to develop even more exciting new modalities. So really big industries around this is in gene therapy, vaccine development, and cancer treatment and diagnostics. And this is also where we saw some of the very early formulations. So as I mentioned, the main quintessential particle when you start off with looking into nanomedicine and nanoformulations are liposomes. So these were developed in the 60s, actually called bangosomes originally by the person that invented them, Bangham. And this didn't sound apparently that well, so they were down to liposomes in the end because they're mainly consisting of different lipids and cholesterol. This was a very interesting growing field of study that eventually culminated in the 90s in the first liposomal formulation that was FDA approved, called Doxil, which some of you might have heard of before. So it's the liposomal formulation of doxorubicin, which is an anti-cancer drug. And this is really where the drug itself, doxorubicin, was very effective, but it had a lot of nasty side effects, as a lot of cancer medicine have. Mainly that I think it was very damaging to the heart. But it was very good at killing cancer cells. So what they needed was a way to shield most of the body from the harmful effects of doxorubicin and get a more specific targeted delivery only to those cancer cells. And the way to do that was to encapsulate that in those liposomes, and there's a very interesting loading mechanism, actually, I won't get into for Doxil. That allowed the very effective drug to be much more safely, accurately delivered and to improve the efficacy of that formulation. But it's a new class of medicine, and it has a lot of regulatory implications, of course, to get that on the market. So it was a while before others came on the market. There's a few of those liposomal formulations on the market, but really not necessarily much more than a handful until recently. And then we had a big next step, which was the first RNA-based formulation, lipid nanoparticle called Onpattro or Partisan, I think, which was developed. And this was really a big breakthrough in the field, again, to show that these kind of modalities with a very unique way of interacting with cells and with the body. This was a silencing RNA, siRNA, that to really be a new breakthrough in medicine. And all this work actually kind of accumulated into the development of the COVID-19 vaccines, mostly by Moderna and BioNTech, which are also RNA-based LNPs, so lipid nanoparticles. This was developed, of course, in record time. Several billion people in the world have had this successfully injected in them. But what we can see now with this field maturing and more particles coming on the market and systems like that, and more pharma being involved, is that there's two big pushes here for new analytics. So it's coming from the regulators, of course, which always want to make sure that these formulations are as well controlled as possible and very, very safe to use. But it's also coming at the same time from all these companies that have these programs, because they see they have issues in the development, which is taking a lot of time and a lot of money to develop, of course, these new formulations. And they have a need for better analytics in this space as well. So many different types of particles that we're using in this space, as I mentioned. So liposomes and lipid nanoparticles are very big ones. And we have polymersomes, or different types of polymer nanoparticles, which are fully synthetic ones, which have a lot of advantages for very tight chemical control that you can have over them, but they're a bit more of a niche product still. And then, of course, using nature's own nanoparticle that's very effective at transfecting cells, such as viral vectors or so lentiviruses or adeno-associated viruses are mainly used here. And these are then loaded with new cargoes, and it's a very different production process. And as well as exosomes, which are cell-derived nanoparticles, which are very interesting from both diagnostic perspective, but also as a therapeutic delivery vehicle. But certainly many regulatory challenges for that one on the horizon. So what I wanted to show as well is just an enormous diversity here in this space. So there's lots of different carriers that are being developed. And this is also because there's lots of different molecules that you might want to deliver, as well as lots of different diseases you might want to target and different tissues you might want to target. So this just naturally happens that targeting to the liver is pretty easy, but everything kind of ends up automatically in the liver unless you very specifically try to redirect these particles to a different spot. And all these particles have a lot of different analytical challenges to them. So more synthetic nanoparticles or particles that we build up from the ground up ourselves in the labs, like lipid nanoparticles, they have very different analytical challenges than natural particles like the lentiviruses. So if you make the viruses in a cell reactor, so it's coming out of, as being produced by cells in that reactor, you have to split them off, and it's in principle a very messy biological process, and there's a lot of purification, for instance, that's required. So for lipid nanoparticles, that's very different, and you can make them much more rapid and much more variety, basically, of formulations, but they need a lot more work in terms of stability and targeting, for instance, that is kind of naturally imparted in these lentiviral vectors or different viral vectors. So other things that people are really interested, of course, in looking at is the composition of the particles. In first instance, of course, is my drug or my nucleic acid loaded correctly in the particles, and is it effectively loaded and in the right spot even? But also this carrier composition is very critically important. So most of the patents, for instance, around the COVID vaccines are really about the lipid compositions and the specific lipid components that were developed to actually make this a very effective vehicle for transfection. And this is something that you really want to be able to analyze as well. Of course, concentration, so number of particles is important. The size of these particles is very important. So these are usually around 100 nanometers or a little bit smaller than that in this space. And that just is the most effective way of getting these particles delivered and avoiding it getting stuck in the body or taken up by the body in the wrong place. Targeting is a very big part of this as well, is different types of targeting ligands that people try to put on, again, to get away from the liver and to other parts of the body. And particle heterogeneity is a really big one I will come back to. So it's very difficult at the moment to actually measure how homogeneous these particles are from each other. So quantification, contamination all speaks for itself, but these systems all have different questions and different possible contaminants that we might want to analyze, and where maybe the conventional techniques are not going to be adequate for that. So just focusing a little bit further on LNPs for those that are not maybe that familiar with that. So this is a structured lipid nanoparticle, and a bit different from a liposome. So a liposome is really a very distinct bilayer, while an LNP can have more complex internal organization, and this gives them or can give them a more stability and more capability of actually entrapping this RNA or mRNA in a more stable way inside the particles. And the crux here is usually around the cationic or ionizable lipids. So the nucleic acids themselves have a negative charge, and these cationic lipids allow them to complex basically with that RNA and ensure that they end up inside the particles as well. And there's lots of other lipids around here that help with the fluidity of the membrane and the structure. Cholesterol is always an important factor here as well. And you can add things like polyethylene glycol on the surface, so pegtails, to make them more stealth-like, to avoid a lot of additional protein corona formation and stability issues in vivo. So how do we make these kind of particles? So again, all these particles are made in different ways, but with LNPs is a very big factor here. There are ways to make them in the lab very easily. You can make them on a benchtop. You don't need even a lot of fancy equipment for them. And there are, of course, commercial solutions available now to make them in much higher throughput and much more control. But in principle, it's something you can make on the benchtop with relatively cheap components and some syringe pumps. So there are some, especially also for liposomes, ways to make them so thin film hydration. You basically make a lipid film and then you rehydrate that with agitation or sonication, and then actually it forms these particles in the solution. Or you can do that while it's mixing. That those don't tend to be ideal for RNA loading. So T-junction mixing or flow mixing is really the key production method here, where you have a lipid phase and an aqueous phase and stream those together with specific channel diameters and flow rate forces to actually force these particles to form together in the solution. And then there's cleanup steps afterwards, basically to get rid of that. And a more sophisticated version of that basically is microfluidic methods, where you can have even more control of how these components interact with each other. So when we're looking at particle analytics, what are we actually measuring? What are we looking for? So the main things are size analysis and compositional analysis, and of course quantification then deriving from that. So what we are looking for is the mean particle size. So we don't want the big size distribution of these particles, but ideally like them all to be around the same size. It's important to detect aggregates as well, both from, you don't want them in there because they are not the effective particles, but if you have big aggregates in there and you try to inject that in someone, that can of course cause issues in that delivery. And deriving from that would be stability of these particles in solution. As you might know, for the COVID-19 vaccines, almost all of them needed to be stored at -80 and then very carefully thawed and shipped. So this is an important measure to make sure that these particles are stable. And for compositional analysis, as I mentioned already a bit, we're really looking also at the carrier composition, the loading, the targeting, and any contaminations in that solution. And then, of course, quantifying how much we have in there. And then techniques that people use for this tend to be traditionally more bulk-based analytics. So this is giving you the average size, for instance, of the particles, or the average composition or loading of these particles. So this is quite a natural development, of course, because this is kits that have been used and techniques that have been used for decades in analytical chemistry labs and adapted basically to look at these new kind of particles. But a lot of these methods, for instance, require the particles to be broken up again and then the contents analyzed. Or if we're doing the mRNA encapsulation, you look at how much basically has not ended up in the particles, or you break up all the particles and look at the contents and then quantify basically the encapsulation efficiency of your drug. But this doesn't say anything about how that is distributed between your different particles. And that's a key aspect of the more single particle-based analytics that I'll get to in a minute. So the advantages of these kind of techniques is that they generally tend to be very robust and very widely available. Our labs already have them. There's a lot of existing expertise around these. And because these are the mainstays of analytics, a lot of the regulatory approvals are based on measuring parameters basically with these kind of techniques. However, the drawback is, as I mentioned, these tend to be bulk-based techniques. You tend to need quite a few different techniques to be able to get to different answers, and they are not specifically designed typically for nanoformulation analysis. But they definitely have their uses, and things like dynamic light scattering, for instance, is very, very easy to use, and you get very rapid information on the size of your particles. But there's a lot more information here, for instance, to be gotten. And the same goes for instance, looking at the RNA components. Ribogreen assay is very commonly used for that, but there's a lot of detail basically lost in that process of analysis. So this is where you can really see, and I would say this is a relatively recent development, perhaps the last 10 years, if not even more recent than that, where people are starting to look for new analytical tools in this space to really get towards the single particle level. So if we're looking at a cargo and actually spending a lot of effort on designing that carrier in a lipid nanoparticle and making custom ionizable lipids and really tweaking those cholesterol lipids ratios to get like the perfect membrane composition for encapsulation and cell delivery, but we then only are able to analyze that in bulk and not actually be able to look on a particle level if that's exactly what we're making. That's of course a really big gap. And what I mentioned in the beginning is really interesting at the moment. It's really currently ongoing development is where there's of course the regulators post-COVID are thinking, okay, there's a lot more formulations coming on the market. We had a lot of urgent demand, of course, for this vaccine because the whole world was in lockdown. But there are perhaps more things that we want to know about these particles and more critical quality parameters that we would like to measure for releasing of these products to the population. And at the same time, these pharma companies, or everyone making them, are thinking, okay, how can I speed up this process? How I can know more about my particles? How I can optimize my processes better? How I can scale them better? and to get to much more effective formulation, because we all know how expensive clinical trials are. So you better know 100% sure as much as you can about your particles before attempting such a stage. So this is basically adapting the slide I just showed into the need for single particle based approaches. And this is really the crux basically of my story here, is that we're looking, the need for this is really driven by heterogeneity. So it's the key word here. So what we could measure before is average particle size, for instance, and particle size distribution. And this gives an indication of the heterogeneity, but really not down to the single particle level. And also being able to link this to the composition and loading of the particles. And for the composition, exactly looking at the variation in composition in your formulation, and the drug loading as well. Is how half my particles empty, for instance, and half my particles have lots of drug inside, or is this all equally distributed? From a bulk-based analysis, you cannot tell that. And of course, everyone understands, I hope that makes a really big difference in vivo when you're trying to deliver this. And these challenges are seen both in synthetic particles or lipid nanoparticles, for instance, or in viral vectors. Same goes for targeting. If you devise a great targeting ligand to put on your particles, you really want to know that that's kind of on every particle in equal amounts, and not that 10% of your particles are completely covered in your ligand and the rest doesn't have that. And this is again linked to different scale up of the processes. So usually you start making a couple milliliters maybe in R&D lab, but if we're trying to scale that towards a production level with completely different methods, different equipment, you really want to be able to analyze that, how that affects your process. Great. So a couple techniques here. There's of course a lot more being developed. I just put a couple here on the slide from different papers as well, things that we've used. So single particle Raman trapping is something that we do. I'll explain a bit more about that in a minute. Fluorescence correlation spectroscopy is maybe a technique that many people might be very unfamiliar with, but I think it's quite an interesting development in the field, as well as advancements in flow cytometry. And then, of course, things like cryo-TEM are very good at looking at structure and also size of your particles, but very expensive equipment, needs tons of training to be able to actually use effectively, and a very, very long time-consuming workflows as well. So very valuable, but not a production-based method, basically, that we can use. And then a particle tracking analysis is kind of an advancement on dynamic light scattering, in the sense of looking at size and concentration. And this is really by looking at Brownian motion diffusion of these particles to figure out their size and their concentration. Very widely used as well. And something I'm not going directly into more detail here because of time constraints, but I just wanted to lift out the FCS, the flow cytometry, and the Raman methods here. So with flow cytometry, you might be familiar with this from a cell analysis. So it's very widely used in analysis of cells in terms of their concentration and labeling of cells. And this has been relatively recently developed into nanoflow cytometry type methods, where this is adapted to be actually able to look at these much smaller particles. So with traditional flow cytometry, it's a dynamically focused flow of single particle stream, and they go through a laser, and it looks at the light scattering and fluorescence signal basically of these particles. And in order to make that compatible with something that's much smaller than a cell, so down to about 40 nanometers, you can make some adaptations there, of course, to the detectors, but also to narrow this sample stream about 10 times smaller, and by increasing the residence time basically of these particles from about nanoseconds or microseconds to milliseconds in the laser beam, because we have a much smaller particle, of course, generating that signal. So it's a very, very fast technique. It can do up to around 10,000 particles or so per minute that you can look at with this technique. And you can determine the size, the concentration, and look at cargo loading, for instance, and surface functionalization of these particles. It's a very powerful technique. I would say the only drawback here is that you need to fluorescently label what you're looking at. So if you want to look at your RNA or targeting ligands or anything like that, you will have to put fluorescent labels on that, and that's the source of your your signal. So fluorescence correlation spectroscopy is a little bit similar in that you need fluorescent labels, but this technique looks at diffusion through a stationary small confocal volume, and we're looking really at on a much smaller scale diffusion of basically single fluorophores throughout this confocal volume. And what you'll be able to assess here is based on the diffusion mechanics and something called the autocorrelation function, is how many of these fluorophores are going through your confocal volume, but also the speed at which they diffuse, which you can then infer the size of these from. So this is really, really good if you're looking at, for instance, doing a targeting ligand on a particle, because the targeting ligand by itself will diffuse much faster than when it's associated with the particle itself. So you can then see if for instance, all your targeting ligands fluorophore signal suddenly goes much slower through the solution. You can see that this is attached to the particles in question. There's lots of other things you can do with this, but also really looking at size, concentration, loading of these parameters. So this, as you can see, together with flow cytometry and what's coming up with Raman spectroscopy, is another advantage of these single particle new techniques that are being developed. It's not only that they're on the single particle level, but also that they're very multi-attribute and multi-parameter. So almost all of these techniques, we can measure many different properties of these particles at the same time because they're dedicated particle analysis techniques. So Raman spectroscopy, for those that might not be familiar with that, is a label-free compositional analysis technique. So what you do with Raman spectroscopy is you irradiate a sample with a laser, and most of the photons hitting the molecules in your sample will scatter elastically as it's Rayleigh scattering. That doesn't give any chemical information, but a very small fraction of that light actually goes to scatter inelastically, which means that it transfers a little bit of the photon energy to the molecular bonds in the sample. And then with a very sensitive spectrocarbon detector, you can determine then how many of these photons are basically how much energy they have lost compared to the laser line, which means then that you can then link that back to which molecular bonds they interacted with. So this allows you to get a chemical fingerprint basically of the sample that you're looking at. So traditional Raman spectroscopy, I would say back in the day, it was a lot of use for inorganic samples as well, and things like in pharma, maybe for solid drug products. But it has really rapidly moved into life sciences. A lot of cell imaging, for instance, is done with Raman spectroscopy as well. And it's moving now into the nano space as well, because of this powerful way of doing label-free compositional analysis. So very complementary to the fluorescence-based measurements, but here the signal is coming from the molecules themselves, and you can look at much more things at the same time. So if we're looking, for instance, at an LNP, you could label fluorescently your targeting ligand maybe and your RNA, but with Raman, you'll be able to see also all the different lipid components at the same time. The big drawback with Raman is that it's much slower. So you will need a few seconds of analysis per particle, basically, to get enough signal from this because it's a spontaneous method. Single particle Raman analysis is very new, developed, and this is really an optimized way of combining this with optical trapping. Sorry, I noticed I'm pressing the wrong button to advance. There we go. So what we do here is we kind of combine two Nobel Prize winning techniques, so optical trapping and Raman spectroscopy. And what we can do is then actually focus a very tightly laser beam into this particle solution and hold one particle in space, and then generate that Raman spectrum of a particle. So that allows us to really make this into a nanoparticle analysis technique. And this can also be automated so that you can actually build up this compositional information of these particles all at the same time. And then this is also a very multi-parameter technique as the others. So we can look at the composition, the cargo loading, functionalization, size, and dynamic events as well throughout this formulation. And this gives you this really powerful information that is missing from the bulk analysis. So really, what is the heterogeneity of the size of my particles, the heterogeneity of your cargo loading, the heterogeneity of your lipid composition, for instance, of these particles, or any contaminants in the solution. Not just the presence of them, but actually are they associated with the particles, which again, if you break up all these particles before your analysis, it's something you're not able to see. So just a little bit of further information on that. So the top is basically my favorite example to show what single particle analysis brings, which is relatively simple to show. So you can see here a small axis of this Raman signal at 700. We have this cholesterol peak, which is very stable, and then these are lipid nanoparticles with different LNP lipid compositions, and you can see the signal basically from this choline group from this lipids go down. And if you had a traditional Raman spectrometer, this is what you would see, and you would see the variation basically is your measurement variability. But with a single particle Raman technique, you can actually look at the variability in that compositional signal on a single particle level. So on the right, you can see every dot here is actually a single LNP where you can measure a Raman spectrum from, and then calculate this ratio. So this suddenly reveals that between these different particle formulations, there's actually quite a lot of heterogeneity in these formulations. And if you can measure that, you can then start adapting your formulation process to try and make this much more effective and homogeneous, rather than going to cells, for instance, and trying that out and seeing a lot of variability and then going back to the drawing board, which will take much longer time. And you can look at Doxil, for instance, as I mentioned in the beginning, and you can see a lot of heterogeneity in that loading, which is very well known for Doxil, that some particles are empty and some have very much in there. And same for viral vectors as well. You can start quantifying populations of low loading and high loading, and this is really the advantage of single particle based analysis. So we're getting to the end here. So the key takeaway of this is that the field seems to be moving towards more adoption of a variety of single particle based techniques, and also the advantage that these techniques tend to provide is looking at many different things of these particles at once, which will surely lead to development of new quality parameters and regulatory approval eventually of several of these techniques, to be able to look in much more detail of these. But the key advantage here is that we're of course not doing that for the regulatory approval. We're doing this also because it will make the process of actually developing these formulations much faster and much more effective, and thus also more cost effective. And this can only be better to bring more and more of these formulations for a wide variety of different diseases on the market in a quicker fashion. So this was my overview of these techniques and the challenges they're in, and several solutions that are coming up. So I'm very happy to take any questions around this. Thank you very much.
Well, thank you, Dr. Penders, for that very informative presentation. Let's go ahead and dive into the Q&A segment now. And we encourage the audience to continue submitting your questions as we dive in here. We have several questions coming in from our audience, so thank you all for being active this morning. Let's start here. And we say, thank you for your presentation. It's really interesting. Could nanoparticles be used not only for delivery, but also for removal of specific particles in specific tissues?
Oh, that's a very interesting concept. If I understand correctly, so the idea was, instead of delivering something to the body, like a drug or something, can we like add particles to draw something out of the body or so? I don't think I've ever come across something like that. Maybe there are certain ways where you could complex, if you make particles, for instance, that would be sticky to viruses or something else that might be in the bloodstream, that could complex. The thing that comes to mind, which is a very interesting and sadly a bit underfunded, I think, field of study, is malaria-based vaccines, which is very difficult to develop. And here, there's actually been several studies that I know of where the goal of the nanoparticles basically is to attack the parasite in different stages and complex basically with that parasite, making it impossible for it to kind of reproduce and it basically stop the cycle of that production and infection of malaria. I think that comes probably the closest to what I've seen relating to that question.
Great, thank you for that. Our next question is chitosan still used as a nanoparticle?
Chitosan, I think, it's not. I'm not super familiar with that, unfortunately. That's not something that I think is. Maybe that means that it's not used a lot anymore. But I'm sure it has specific applications. But that's not something I'm very familiar with, unfortunately.
Well, maybe that can be something to take offline with that particular question. But thank you for giving it a go here. We have a few more. Let's move on to this one here. How can you prevent the immune system attacking the particles?
Right. So this is something that is done very experimentally. I think by trying to modulate that in first of all, of course, on in vitro and then in vivo type studies, is that specific components tend to be more agnostic, basically, to the immune system, or are not specifically targeted by the immune system. And this is also where this stealth aspect comes in, where the pegylation was discovered to be much better at actually masking it from the body's natural response. There's an interesting concept, though, with the vaccines, is where you definitely need certain level of immune response to actually make the particles effective. So there were actually COVID vaccines developed that were too invisible, basically, for the immune system, which then doesn't actually trigger the right cascades to actually bring that immune recognition of what you're trying to deliver, or to get that going. So it's a tricky balance to tread. Yeah.
Understood. Thank you. Let's see here. What is the name of the dye used in flow cytometry for nanoparticles? Is it multi or one for all nanoparticles?
There's lots of different dyes that are being used for that, and there's lots that came from standard fluorescence microscopy, which of course has been around for a very long time around cells. But there's also specific dyes being developed especially for nanoflow cytometry as well. So one of the challenges, for instance, is that if you need to label your mRNA, for instance, before you can encapsulate it into the lipid nanoparticles, then in order to measure it, to be able to see it on the flow cytometer, then that labeling of your RNA can actually affect the RNA encapsulation as well. So you're then studying kind of like a proxy system and not your real system. So, and I believe what's been developed is dyes that can actually penetrate the LNPs to be able to add the dye after the particle has been formed, and actually then label the RNA in that sense. So there's always some control experiments, of course, that you need to do there. So if you had non-specific interactions with your dye and your particles, that it could flag up as RNA signal, but it's not actually RNA signal. So you have to be careful with that. But there's lots of different dyes there for different type of particle systems that you can use, and also different colors. So it's called multiplexing, right? So usually you tend to maybe have like a blue fluorophore and a green one and a red one, or there's lots of variations in there. And then you can look at multiple at the same time. So you could look at cargo, for instance, and targeting at the same time. But it will kind of be limited usually to a couple of these. So you can't do 10 different dyes or so. The flow cytometers won't be able to handle that with the detectors, typically.
Great, thank you for that. We got one more in here. What nanoparticles are used these days?
For this is of course a very wide field. So I would say that we definitely see a bit of a shift from liposomes to LNPs because of the extra stability they afford to make, and also they're very easy to make with different techniques. So I would say that liposomes tend to be maybe a bit less used now with the advent of LNPs. But what's actually, I find that the most exciting thing about this field is not just a one-hit thing. And there might be some companies or some people that think, okay, once we have our LNP shell, like the COVID formulation, for instance, and we can just swap out whatever is in there, and there will be be effective, but that doesn't really tend to be the case. So the fact that this field actually has such a broad variety of different particles to target all sorts of different tissues, deliver all sorts of different molecules, is really what makes this very exciting, also from an analytical perspective, of course.
All right. Let's move on to a few others here. What are the advantages of Raman spectroscopy, based analytical approaches?
So I would say the main advantage of Raman is that that's label free. So it has basically no prep required. You tend to just dilute your sample to be in the right concentration range for the machine that you're using. And then there's none of that labeling or dyes is required for the fluorescence-based techniques. And the other aspect of that brings with that is that which I kind of call like holistic analysis. So if you're labeling fluorescently, for instance, your RNA, you're specifically looking for RNA content, right? That's going to be your assay to be able to look at that. But there might be other things going on with those particles that you're not necessarily thinking of or don't have a way to also measure all of that. Like no one is, as far as I know, fluorescently labeling cholesterol to look at the cholesterol content necessarily in these particles at the same time. And with Raman, because you don't need to do that labeling, you can get that kind of information, that maybe the information that you weren't specifically looking for, but might be very important to know about your formulations. I think that's the advantage. Disadvantage, as I mentioned already during the talk, is that Raman is a much slower technique. So whilst with the flow cytometer, nanoflow cytometer, you can maybe chuck through 10,000 particles in a few minutes. On a single particle Raman machine, you're more thinking about analyzing a couple hundred in an hour. So it is a very different scale, which is something you have to take into account when you're doing your data processing, of course.
Great, thank you. It's another one from our audience. Are there any diseases where nanoparticles are currently used to deliver drugs?
So I would say the biggest probably for drug delivery is cancer. So different cancer modalities tend to be quite heavily using the nanoformulations. This is similar to the example I gave with Doxil. So it's quite easy to kill cancer cells if you have them in isolation, right? So there's lots of chemicals that are excellent at killing these cells. However, the main problem is that they're also excellent at killing all the healthy cells. So you really you're kind of killing the cancer cells just a little bit faster than the rest of the body. And that's why there's so many side effects to all the cancer treatments. And nanoformulations are one way where you can try and shield the body from the harmful effects and try and target only the cancer cells. So this is really where a lot of nanoformulations are being used. And also things like antibody drug conjugates, antibody-based therapies like Herceptin, for instance, for HER2-positive breast cancer. Very effective way of actually targeting these types of cancer.
Great, thank you for that. We have time for just a few more here, so let's go ahead and move on to this next one. What are the differences in challenges faced by manufacturers between widely varying particle types, such as LNPs versus viral vectors?
This is a challenge because those production processes are completely different on a completely different scale. So with an LNP company, they can have different off-the-shelf machines as well that can make formulations, and they can make 20 formulations a day or 50 formulations a day. Like it's very, very rapid to do optimization and make different formulations. While if we're looking at viral vector manufacturing, because it's a cell-based process that can take many days to make one batch. Of course, you can have multiple reactors at the same time going, making different formulations, but it's a much, much slower scale. And for LNPs, therefore, is the need for automation of these analysis is much, much greater. So you have a much higher quantity of different parameters to test. And we want to look, for instance, that there's very few contaminations. Can be a very clean process. The main thing that people look at is residual ethanol from that lipid solution to look at, and maybe some leachables from the tubing. Whilst with viral vectors, it comes in a soup basically with the viruses and the cells and all sorts of proteins and nutrients, and so from that reactor, and it all needs to be very carefully cleaned up from these particles. So there's a lot more purification detection needed there. So this is, and then having techniques that fit for both is quite a challenge. Yeah.
Great, thank you. Let's move on here. What do you think the future of analytics of these gene therapies looks like in 10 years' time?
Yeah, so it's always a tricky one to predict, because 10 years sounds like a long time, but if you look 10 years back, it's not that long, of course. But I think, well, it's quite obvious from my presentation that I think it's moving towards more single particle analytics versus bulk-based. But I think in the single particle analytics, what we're seeing is much more automation. So, and this goes beyond just being able to preload samples and hands-off run 10 or so overnight or in a couple of hours. This is also really moving towards process analytical technology. So inside the machine, basically, or coupled to the machine that you're actually using to make it, or the bioreactor where you're actually making your viruses, to have the analytics integrated into that directly. So to move away from making a formulation, taking that sample, putting it on an analyzer, separately in a different location or different lab, that's all coupled together in that production process to have even more automation there. And then, of course, there's a whole world of software and possible AI tools and so that you could bring into that as well to really make it as hands-off and automated as possible. I don't know if that will all happen in 10 years, but certainly a lot of people are trying to go there.
Indeed. All right. Well, we have time for just one last question here, and that is this. What are the advantages or limitations of Raman-based analytical approaches versus fluorescent-based assays? So
I think we kind of covered this in a previous question from the audience, which was really based on, this rapid single or small amount of detection based on fluorescence versus a slower detection method that can look at many more things at once in this holistic type of analysis. So I don't see them as exclusive to each other. I see them as very complementary to each other. But this is some of the things you need to look at when deciding on which analytical techniques you're using for answering what question.
All right. Well, that wraps up our Q&A. So thank you again, Dr. Penders, for your time today and for your important research. Now, any questions that we didn't have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you registered with. This webcast can be viewed on demand for 1 year until October 16th, 2025. And LabRoots will alert you via email when it's available for replay. We encourage you to share that email when you receive it with your colleagues who may have missed today's live event. Thank you again to our audience for joining us today and for your important and wonderful questions. And we encourage you to continue to explore this incredible virtual space and to join us for the other scheduled presentations coming up. Until next time, everybody, goodbye.